Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Azhar Rafique, Assistant Professor, Department of Zoology, Government College, University, Faisalabad. The topic of my presentation is efficacy of anthelmintic properties of leaf extracts of Moringa oleifera and as a director indica against nematodes of ovis orientalis. First of all, the introduction. Helminths are the parasitic worms that feed on a living host to gain nourishment and safety while inflicting terrible nutrient absorption, weakness, and disorder within the host. Sheep gastro-intestinal parasites are of public health significance and they are incriminated in zoonotic transmission to human. Buffalo sheep is an animal species that is widely hunted and produced in Central Asia. Buffalo sheep are very sensitive to many parasitic diseases, especially fasciolosis, nematodiosis, etc. These diseases cause health problems and animals die prematurely and cause economic losses to breeders. Chemotherapy is a chief remedy modality used for helminths infection in animals. However, because of increasing improvement of anthelmintic resistance, phytotherapeutic tablets are secure, non-poisonous, biodegradable. The consequences recommend that the phytochemical drugs for using diverse illness. Moringa leaves and stems contain amounts of bioactive compounds. All parts of the neem plant, including the leaves, bark, fruits, seed, oil, and sap, have been shown to have medicinal properties and contain over 10 different active components with azadirachata as the most potent and widely studied. Then the materials and methods. And the preparation of, first of all, the preparation of plant extract, collection of leaves of Moringa oleifera and azadirachta indica, then dried at room temperature, ground into fine powder and extracted with aqueous methanol and ethanol solvents. Egg hatch assay. Aqueous methanol and ethanol extracts of both plant Moringa oleifera and azadirachta indica were used as the test treatment. Fecal samples of meflon sheep were collected at the Wildlife Park Gatwala, Faisalabad, Pakistan from the ground after defecation. Approximately 9 to 110 eggs of nematodes in 0.5 ml water placed in each test tube. Serial concentration of each plant extract in total volume of 0.5 ml in distilled water was added to make concentration of 50 to 1.5 milligram per ml together with water containing the eggs. The test tubes were then covered and kept in incubator at 27 degrees Celsius for 48 hours. The experiment was conducted in duplicates for each concentration and replicated 3 times. Hatched larvae, dead or alive, and unhatched eggs were then observed under dissecting microscope at 40 multiple magnification. Then the larval test, development test, the experiment was conducted in plastic cup of 20 ml according to the technique described by Costa et al. An aliquot of 1 ml containing 100 to 121st stage larvae of nematodes were mixed with 5 grams of feces from a sheep free of gastrointestinal nematodes and 6 serial concentration of the plant extracts with water containing larvae and volume of egg free of feces. All samples were incubated for 7 to 14 days at room temperature. The experiment was conducted in duplicates for each concentration and replicated 3 times. Then 1 drop of Ligol's iodine solution was added and all L3 stage larvae were counted under dissecting microscope at 40 multiple magnification. For positive control, ivermectin 1% was used while negative control, untreated eggs and water were used for both tests. Then the results, this table shows the egg inhibition, egg hatching inhibition percentage and LC50 of 2 plant extract compared to negative and positive control. And the all plants showed egg hatching inhibition by increasing concentration of extract. The egg hatching inhibition also increased. The maximum egg hatching inhibition was 88% in ethanol solvent of Moringa oleifera, while the maximum concentration of egg hatching inhibition was 94% in methanol solvent of the azadirachta indica. These are the values of the LC50 of Moringa oleifera in aqueous methanol and ethanol solutions. And these values were the azadirachata indica in aqueous methanol and ethanol solvents. This table shows the development of the larval inhibition percentage and LC50 of two plants extract compared to negative and positive control. Both plant extracts showed larval development inhibition. The maximum inhibition was observed in methanol as a solvent of azadirachata indica. These values were LC50 of Moringa oleifera in aqueous methanol and ethanol solution, and these values of LC50 of azadirachata indica in aqueous methanol and ethanol extract. It was observed that methanol extracts of azadirachata indica is the most effective against the nematodes. Then the conclusion from the present in vitro study, it is concluded that ethanolic extract of Moringa oleifera and methanolic extract of azadirachata indica exhibited significant anthelmintic activity against nematodes of sheep and have potential for use as an alternative anthelmintic agent for the control of gastro-intestinal ailments of wild sheep and other ruminants. The two plants in our study were effective in the treatment of gastro-intestinal nematodes of wild sheep. However, it would be necessary to conduct in vivo parasitological studies to consider the metabolism of extract in digestive tract of ruminants in the captivity. Thank you for your attention.